Great. Okay. So yeah, I have my screen with the slides here, and here is you. So if you see me doing like this, it's because I'm looking at you and looking at the slides as well. But yeah, today I wanted to share with you a little bit of a journey we had when we decided to build a large-scale segmentation data sets of X-ray images, and maybe share with you some of the lessons we learned through this journey, right? Nowadays, I think many people are working on building new data sets. In our case, as I will tell you afterwards, you will see that we build this data set on top of existing data sets. So we kind of augmented the data set. And that's the story I want to tell you today. Because, I mean, we all have seen the importance of these large scale data sets, right? Like even from ImageNet for computer vision, that was a huge breakthrough that happened many years ago already. And we see that every year at MICAI, for example, with the Grand Challenge or in the Grand Challenge repository that we have, where basically people are launching these huge data sets that are fueling the development we are doing. Nowadays, we do AI, many of us, I guess here, we do AI for medical imaging, and working without a data set, it's really difficult. Like it's difficult to do something without data, no? So that's why during the last years, I've started to look at data sets themselves. Actually, this is an image that I liked a lot from a really nice paper from Amalia Jimenez Sanchez and her collaborators that I really recommend you. Actually, during my talk, I will be pointing to some papers that I think are interesting in this direction of creating data sets, building new data sets, and what are the issues that emerge when doing that. And when we think about a medical data set, well, of course, we are basically a medical imaging data set as we are working here. Of course, we will have images, we will have metadata, we will have labels, and we will have a license associated to it. That's the anatomy that Amalia was kind of defining in this paper, and I really relate to that. Today, we will be focusing on the labels part. Basically, the data set we will be creating is mostly about labels, but these other aspects of the data set, the images themselves, the metadata, what's, I don't know, the patient information that we included, and the license, I will tell you a little bit about the licenses as well, are also really important aspects that need to be discussed. If you are interested on this topic, I really recommend you going to this paper that we are presenting this year, in fact, together with Amalia. Actually, when we discussed all the issues that emerged when creating this dataset. Actually, this was a nice paper that came out of a workshop we organized in Copenhagen last year. And many of us wrote there the issues that, I mean, I cannot cover the whole paper here, so that's why I'm pointing you to it in case you want to have a look at it. But today, I will be talking particularly about a dataset we released last year that is called CheXMask. And this is a large-scale dataset of anatomical segmentation masks for multi-center chest X-ray images. And what does this mean? What does it mean? Sorry. Well, it means that we have images that were already there. So this was a public, like there are many images of X-ray going around the world. Like we have, for example, the CheXMask dataset, or we have the Montgomery dataset, or the Shenzhen dataset. There were all these image datasets, but these datasets, they didn't have segmentation labels. And as I will show you in a bit, we had some models that were working pretty well for multicentric segmentation. So we thought about creating a new dataset that is basically a lot of segmentation masks for these existing images. Okay, that's the setting in which we will be. So what we released with this dataset were about more than 600,000 segmentation masks for the heart and the lungs. And what was also interesting about our dataset, I think, is that these were not only segmentation masks at the pixel level, but these were segmentation masks at the node level. So it's what we call like landmark segmentations. We had correspondences in the segmentations we are releasing, the different parts. So you can see, actually, you can look at the contour of a segmentation as a graph, and these are going to be nodes along the contour of the segmentation. And what you will have are correspondences in the sense that this node here is always going to be called 21, for example, or this node here in the upper part of the heart is always going to be called 10. So you can basically do some kind of shape analysis on these contours and get information about anatomy and in different patients and the different conditions and the different diseases. And I think the interesting thing about our data set, and one of the problems I'll be discussing today, is that, okay, we had the images, we generated the segmentation masks, but these were more than 600,000 segmentation masks. So the question that was interesting to us was, how can we provide some kind of automatic control metric per subject, a number that tells you how good is this segmentation mask that we are providing to you? But we had to do it automatically. We cannot ask a doctor to basically label or check 600,000 segmentation masks. So a little bit of the story I'm going to tell you today goes on how we did that. And of course, we validated these automatic quality control metrics that we provided with some expert annotations. So I will tell you a little bit of that. And we tried to do some kind of fairness analysis, basically analyzing these metrics per subgroup, seeing how our segmentation masks are good or not for different subgroups like male, female, young, older patients, and this kind of analysis that is of interest to us when we are thinking about the fairness and how fair our models are. So this is a little bit of what we are going to be covering today. And let me start our journey to create this data set with the data preparation part. So basically we had five different segmentation data sets, image data sets. This was the CheXMask X-ray 8 data set, the CheXMask data set that was released by Stanford. This first one is also known as the NIH data set, the Mimic CXR dataset, the Patches dataset, and the BINDR CXR dataset. And now we are extending it to other datasets as well, but this was the initial kind of bunch of datasets we used. And so we basically defined some inclusion-exclusion criteria. We took only images from the front view, okay, the frontal view of the dataset, and we basically filter by that, and this ended up being these more than 600,000 images that we used. We had to do a lot of stuff, and this is something important when you are working with the creation of this kind of multi-centric data sets, even if you are creating it yourself, creating your own images, like capturing your own images, or using images that come from existing data sets. We had to do some image standardization, so that was fine, that was okay. But then something that was of very relevant to me was this assessment of existing metadata. So we had to harmonize a little bit what metadata was available for every data set. Some of them had, I don't know, sex labels. Some of them also ethnicity. Some of them didn't. So we kind of organized that. And this is the first point where I want to stop a little bit here and reflect in the creation of the data set. And it's the licenses that these datasets had. So as you saw, these were 5 different datasets coming from different parts of the world. So, and everyone was released with their own dataset, like a Creative Commons or maybe like different type of dataset, of licenses. And when we started working on that, we segmented the data sets. We saw our segmentation masks were great. We were really happy. So we say, okay, let's put it together. We wrote a manuscript. We submitted to a journal. And when submitting to the journal, the editor comes back and says to me, hey, do you have the authorization of creating this segmentation mask from all these images? And I was like, okay, I'm not releasing the images. I'm just releasing the segmentation masks. And they were like, yeah, but if you are creating a derivative from these images, you should have the acknowledgement of every one of the authors of these other papers, of these other data sets, telling you that you can release information that derives from there. And this is the first lesson I kind of want to tell you, like, be careful with that. After that, I had to email every author, and every author gave me the consent, and that was great because everyone was really collaborative. But if you are thinking about augmenting data sets and then creating a data set based on that, that's a really important part that touches on kind of the future AI guidelines as well, that we created with Karim and the team. So first point there, and really important. I mean, it ends very well because everyone gave the consent. Then after that, how did we create these segmentations? Well, we have a model that is called hybrid GNet. I'm not going to enter here into the details of the segmentation model itself due to mostly to time constraints. But basically here the idea is that this is a network that we call hybrid GNet because it goes basically from images to contours. So it's not a segmentation like a classic segmentation model at the pixel level. It's not that you are going to enter an image and you will get a segmentation mask at the pixel level, but you are going to enter an image and you will get as a result a graph. And this graph will represent the structure, the anatomical structure you want to have. And you may think, why do I want to have that? Well, honestly, that helps a lot with the robustness of the method. This is if you look at it, it looks kind of like a unit architecture, but it basically has what we call classic convolutional blocks here in blue, like just residual CNNs. It goes to a latent space, kind of a VAE, a latent space. It's basically probabilistic from where we can sample. And then the reconstruction, the decoder step, it's convolutions on graphs. So we basically have a generative model of graphs as a decoder and kind of an encoder that gives you an embedding from an image. That's why we call it hybrid GNet, because it has like classic convolutions on the one side and graph convolutions on the other side. And we created this model that is very, very robust with respect to different changes. Like, for example, if you look here, this is the images we are trying to segment. This is the ground truth you would be expecting to get. And these are different results you are getting with, for example, classical NN unit trained on the same data set, or the classic unit. And this is the results you are getting with our model. And what you can see is that the nice thing about the hybrid GNet is that it is really consistent in terms of anatomy. So you get segmentations that are very consistent. And if you look here, for example, where you have this peacemaker here, well, you will see that our model is including that in the lung, while other models are not including that in the lung. And that's because our model has a strong anatomical prior. It may be useful or not, depending on the type of task you are solving. But for us, we wanted to have what we call the complete masks of the lung, and our model is very good at that. And that's because it imposes very strong priors based on these kind of generative models that we have on the decoder part. Again, I'm not going to enter into the details here of how this model works, but if you are interested, you can check our paper that we published a couple of years ago in TMI. And yeah, there you have all the architectural details for that. So, okay, we had the images. Now we generated the segmentations using our model for all these different centers. And now we want to go and basically estimate the quality of these segmentation masks for every predicted segmentation. Oh, a detail I forgot to tell you here is that I was telling you I go from images to graphs. And so to create this basically pixel level segmentation mask, the only thing we do is to fill this structure in with pixels, right? So that's the way we go from graphs to pixel level segmentation mask, if you go. Okay, so we have this pixel level segmentation mask. We want to now see what's the quality of that. And in order to ensure the quality of these more than 600,000 images, we needed some kind of automation here. The first thing we did, it was to validate it with validate some of these segmentation masks that we created. We took randomly 30 segmentation masks per data set. So we had about 150 segmentation masks. And we asked expert physicians from Hospital Italiano de Buenos Aires, that is one of the best institutions here in Buenos Aires when it comes to healthcare, to basically perform manual revision and check if the results were correct or not. Of course, we then use the segmentation masks that were manually checked to estimate the dice coefficient and the Hausdorff distance, which are two metrics that we usually use to analyze the quality of the segmentation masks. And we could see that for the different data sets we had, the dice coefficients we were obtaining were pretty high. So we were happy with that subset, but that was only a subset of the segmentations that we have generated. That tells us that since we randomly sampled that, so okay, that makes sense. It should be a good quality that we are providing in segmentations. But still, we don't have one estimated dice per image in our dataset. That is what we wanted to have. So in order to generate this per image dice coefficient without ground truth, we resorted to a paper that was published a few years ago by Ben Glocker and his team at Imperial College that is called reverse classification accuracy. And this is a very simple technique that I will quickly explain you now that allowed us to estimate these dice coefficients for every image. And the idea goes as follows. Look, you have your image here, image I, and you have a prediction, maybe the prediction that we generated with our model, okay? But we don't have ground truth, and we want to estimate the quality of that segmentation here as I. Now, what we have is a data set of similar images with the corresponding segmentation masks. And this is ground truth, manual annotations. This we had because we had a few segmentation masks even in our training data set. So you have your image and you have your reference database that we will call. And what you will do is to basically take this image and this segmentation and use it to segment the images you have in your database. So there are different ways in which you can do that. The easiest way is by doing atlas-based segmentation. That means I take this image, I register the image in a deformable fashion, okay? I do deformable registration to warp this image to look like this one. And I take this deformation field and I transfer the labels, okay? I deform the labels in the same way. And then I will have, for every image in my reference database, I will have one segmentation. And what I can do is basically to compute the ground truth between the segmentation I just generated based on this one and every one of the images with the ground truth segmentation in my reference database. What's the intuition here? What's the hypothesis? Well, the hypothesis is that if this segmentation here is good, if this is a good segmentation that I produce, then it will be useful to segment at least one of these images that would look similar to the original one. If that happens, then it means here I will have at least one high dice value. So what I will take as my final estimation of the dice value is the maximum or the mean. You can go with one or the other one, but we usually take the maximum, but sometimes the mean, of these values that you estimated per image. And this is going to be the estimated dice for this segmentation here. And with that technique, we went and we basically segmented a lot of images for which we had ground truth so that we could validate whether that was working or not. And this is the kind of plot that you get when you take the maximum RCA estimation. So RCA goes for reverse classification accuracy, that is this technique here, or the mean. And you can see that both work pretty well. And yeah, maybe the mean has it's a little bit more robust and has a slightly higher person correlation coefficient there. Great, we have now our way of estimating a dice coefficient. Now, if you look at our data set, what you will see is a distribution like this one of dice coefficients, estimated dice coefficients, because remember, we don't have a way to really say this is or this is not the dice coefficient because these are more than 600,000. So here you can see that images that have a pretty low dice coefficient tend to be bad segmentations, while images that have higher dice coefficient estimated tends to be pretty high. And these are the values that we reported in our dataset. Actually, what we did, it was to empirically analyze these images, and something pretty interesting that we found was that the estimated dice coefficient was kind of an outlier detection algorithm, because if you look at images where our model was providing a very low estimated reverse classification accuracy dice, you were finding these kind of images. Even cell phones were there in these data sets. And that's another thing that comes as a learned lesson, is like when you are working with public data sets, be careful with what is there, because you may even have cell phones or images of a leg in a data set that claims to be only chest X-ray images. And the nice thing is that with our method, we could detect those. And so if you go now to CheXMask to our repository, you can filter out all the images with low dice, and that means these images most likely are kind of out of distribution cases. That was kind of the second validation strategy we used to create our dataset. And then there was a third one, a third validation strategy, that it was simply, okay, we can use the segmentations we estimated in the whole, for example, CheXPert dataset, to train a new segmentation model and then go back and segment images for which we have segmentations. And in that way, it's like taking this RCA idea to the population level. We will kind of estimate a dice coefficient for the whole population, for the whole dataset. We will be using it to train a model and then segment back our images for which we have ground truth. And we did that, and that provided, again, very high dices, meaning that the segmentations we are producing for these datasets for which we don't have ground truth are of pretty good quality, because when you segment images for which you have ground truth, you get this kind of very high dice coefficient here. So with that, we were happy. We had kind of 3 different ways of validating this bunch of segmentations that we generated and we released. But there was something else that we wanted to look at, and it's how fair our segmentation model was. This quality that we are estimating, is it useful to tell, hey, be careful because there is a subgroup in which the model is failing more than in the other ones? And that comes down to the fairness of this model, basically. And this is a topic I've been pretty involved during the last years. We started working on that when we published this paper back in 2020, I think, about how having highly unbalanced data sets in terms of gender in this case may impact the performance of classification models. And then we wrote this article as well discussing the impact of this type of imbalances and other reasons for having unfair models. And by an unfair model here, I will mean a model that has a lower performance in one subpopulation than in the other one, a systematic lower performance, right? A model that is failing much more on female than male, or these type of things. And all this discussion on what it means to be fair in the context of medical imaging is on this paper on Nature Comms that we published a few years ago as well, and you can go and check if you're interested. But of course, we were not the only ones talking about that. Actually, many people were discussing fairness of AI in medical imaging, and there was this paper that caught my attention a couple of years ago from Andy King from King's College and Esther Pujol, and they showed that this type of imbalances in the training dataset may not only affect classification system, but also segmentation systems. So we showed that classification system was systematically affected. I mean, I'm not going to enter in the details there, but they showed that, for example, for hard segmentation, you could have very different dice coefficients when you analyze for white, Asian, Black, Chinese population. You can have very different dice coefficients for all of them. And that related pretty much with the underrepresentation on your training data set. And honestly, this is something I was not expecting. I was not expecting that there were so big kind of signals or changes in the different ethnicities when it comes to segmentation, okay, of the structure of the heart. But this happened. So what we wanted to see, it was basically if we could analyze the same kind of fairness analysis for age or for sex in our data set. But the problem is that when you try to audit for fairness in a data set, how do you do that? Well, you usually have some kind of segmentation model that you want to audit. You take the different subgroups, in this case, for example, male and female patients, and you generate a bunch of metrics that are usually based on the gap on performance between one group and the other one, right? Now, like for example, the DICE gap. Now, we need ground truth to do that. We need ground truth to check, okay, what's the performance on this group? What's the performance on that group? So if I want to analyze fairness in the absence of ground truth, how do I do that? How can I estimate if my model is fair or not without having any kind of ground truth in that population? And that's important because if you think about it, and that's a really interesting paper that came out from the group of Jessica Schroef and her coworkers at Google, I think, a couple of years ago as well. What you will see is that fairness does not always transfer across distribution shifts. And what that means is that the model that was fair in a certain hospital, maybe you go to a new hospital and now it's not fair anymore. There are big gaps in performance. And they show that for in these cases for models for skin condition prediction, basically cancer prediction in skin in Colombia. Sorry, and they went on a data set from the US to Colombia, and basically they observed that in the source data set, the model was fair, for example, across different age groups. But when they came to the new target data set in Colombia, it was not fair anymore because there was a lot of, like, the performance on older people was much lower than on younger people. So that can happen. So in our case, we were worried about that. And so the question came of how can we anticipate bias for segmentations in new populations without ground truth? And we call it unsupervised bias discovery, basically discovering biases without supervision. And that was a paper that we published also a couple of years ago at the Fairness of AI in Medical Imaging workshop, FAMI, that we are organizing this year again, by the way. So if you're interested on these topics, have a look at the FAMI workshop. The call for papers is open, and we are receiving submissions, but this is a paper we published there. And where we basically showed, I'm running out of time, I have only 1 minute, so I will go pretty quickly here. But we basically showed that the RCA estimator that we had used before was useful. Like, we did a lot of synthetic experiments and also real experiment to show that if you want to use RCA to estimate gaps in performance between different subgroups, that's a useful tool you can use for that. And again, you can see that this is the real gap in DICE coefficient that we were finding between the subgroups. And this is the estimated one using RCA. And you can see that it correlates pretty well. And actually, when going to real data sets, we observe the same. Again, I'm not going to get much into the details here, but what we use from this study in our CheXMask paper was, okay, let's look at the distribution of RCA coefficients across different subgroups. For example, when looking at, I don't know, sex here, well, we were not seeing big differences. Look, this is on pink here, or orange, you can see for female. And on blue there, you can see for male population, the distribution of DICE coefficients, right? And there are no big differences, honestly, between these two. So we can say, okay, the model is pretty fair in that sense. Now, where we observed very big differences was, for example, here. When we stratify our data by view position, what that means is that we are taking AP or PA positions, X-ray images, like images taken from anterior-posterior or posterior-anterior, which for the eye doesn't have a huge difference. Now, what we observed was that images coming from AP, anterior-posterior here, had a much lower DICE than images coming from posterior-anterior. And the interesting thing is that when we show these results to the doctors, from Hospital Italiano de Buenos Aires, they told us this makes complete sense because AP images tend to come from hospitalized patients, patients who have a lot of maybe cables or artifacts on top of the chest because they are lying on the bed at the moment the image is taken. While images coming from PA are usually patients that just go and take the image and go back home. So the patients from AP are much more difficult, okay, because they are not a standardized view. They have a lot of stuff on top. So it's likely that the model will fail more. And so this is one of the images actually that we illustrate here. It was not sex, sorry. It was not even age. It was mostly related to the view position in which the image is taken, what was dictating whether the quality of the prediction was larger or not. And well, for example, for chest X-ray, there we yes observed that for younger patients, we had a distribution of estimated dyes that is worse than for older patients. And that makes sense also because this data set does not have, I mean, in our data sets, we didn't have a lot of pediatric patients. So if you are going to use CheXMask segmentations, or even our segmentation model, well, is not very good at segmenting pediatric patients as well. So that was the way we found to navigate fairness, to try to understand fairness in the absence of ground truth. That to me is a very real situation. It may happen a lot when you go to a new hospital, that you will have a new population, you will deploy your system, and you will not know how well the system works. Well, these are some techniques you can use to try to estimate that. This is the paper of CheXMask that was published, as I was mentioning, in Nature Scientific Data last year, and you can access it. You can download our segmentation masks. People have been using them in many different studies that we didn't imagine. That's the nice thing about releasing a data set. Studies that go from training mask autoencoders to others that are more clinically oriented. So that was pretty interesting to see. And yeah, we are thinking about keeping on augmenting our data sets. I think augmenting data sets with new annotations is important because it provides new uses for the data sets. So trying to add more annotations about metadata, OOD, and even new anatomical structures is something we are working on now. So yeah, just to conclude, we discussed this journey about the creation of the CheXMask dataset, and we basically show how we could provide automatic QA metrics and validate them using expert annotations, but also automatically, okay? And these metrics we show that can also be useful as surrogates to basically estimate fairness without ground truth. So yeah, that's all from my side. I want to thank the institutions that were supporting this research. Yeah, and I'm open for questions. Thank you.
Thank you, Enzo. That was a great talk. Very impressive set of works. We'll open now the floor for questions. And as always, I will start. So please write down your questions in the chat. My first question is regarding the first part, and I was curious to know what do people think of data sets that are annotated in that manner using sort of a certain level of automation as opposed to fully manually annotated data. Do they still trust them? Do they think this is the annotations of sufficient quality and so on? What do you think?
Yeah, that's a very, very interesting, and that's something we were actually wondering a lot. And it's something we discussed a lot in this workshop I was mentioning on medical imaging datasets we organize in Copenhagen. So of course you cannot trust these automatic labels as much as you would trust the labels generated by a human. But that's why we put so much effort here on trying to validate these metrics. And I think there is the key, providing empirical validation that the metrics kind of make sense. And of course, you have to be aware of that when using the data set, that segmentations are not perfect, that even this metric we are providing per image is not perfect. Maybe you will have an image with a high estimated dice, and maybe the image at the end of the day was not that good. But nowadays, in the era of huge data sets where we need that to train models, well, if you are using this to train a model for segmentation, for example, most likely, and as we showed, the segmentation model you will get is pretty good. So for that kind of uses, then it's okay. I think it really relates to the application you are considering, right? And also, I mean, the application, and that's something important also from our paper. There is a limitation section where we kind of talk of these things. So when you are using a data set, you have to think about that. But think about, for example, the the NIH data set, one of the first ones releasing a large-scale X-ray data set with annotations for diseases. So they had labels like atelectasis, cardiomegaly, pneumonia, these kind of labels. These labels were automatically generated from analyzing NLP reports. Okay, and that was done even before the era of LLM, so that was done with really basic methods for analyzing that. And that data set was not perfect at all. There were many critiques on the labels that we had, but this data set opened so many avenues of research after that. It's one of the most cited papers, I think, in the medical imaging literature. So you can, like, we know it's imperfect, but depending on the application, it may be useful. And you have to be aware of the limitations of the data you are using. And we, as we build these data sets, have to do everything we can to validate the quality of the augmented data sets or the augmented labels we are providing as well.
Cool. So I'm going to move to the question that we received. So there is a question here also related to stage, to the first part. And in general, there was a question on how developers who are not particularly or not always focused on creating data sets, they tend to use existing data set. How can they learn and improve their skills in creating data sets in general?
Well, I mean, this is the main data set we have created so far. It's not that I've been creating data sets all my life, right? So I don't know.
Yes.
And so I haven't worked, for example, on capturing the images. This is another aspect where most likely you will have to involve medical doctors, and you will need, like, here we also involve medical doctors, but for that particular one of capturing the images, there is a lot more to do there, right? So I guess there is a community of people thinking about creating data sets. Like I was mentioning, Veronica Chepligina, for example, one of the people leading that work in Copenhagen. The paper we publish now at FACT that talks actually about these issues that emerge when you think about a new data set, what to consider from legal aspects to shortcut aspects, the type of shortcuts that can appear when labeling. So these are resources I think that people can look at. And now we have even journals about data sets. I mean, we have the Nature Scientific Data Journal, but we also have the data-centric machine learning data set, journal associated to JMLR, and many others. And even tracks in conferences that are devoted to data sets, like NeurIPS has the dataset track, right? So there is a lot of people thinking about that. My advice is go and talk to these communities.
Do you think,
yeah.
Enzo, do you think developers should learn to create data sets, or this is not their job?
I mean, I think it's up to everyone what's interesting. I mean, what's interesting for them, right? I'm a developer, I'm a computer scientist, and I'm interested on creating data sets. But some of my colleagues, they say, why I would be interested on that? And it's fine. They are mostly focusing, I don't know, on new regularizers or stuff like that, or training models.
Yes.
So it's a different aspect of research nowadays, I think. It's important to do it seriously.
Yes.
And yeah, because it's an artifact that we need now, like especially in the era of LLMs. We now release another data set actually for for evaluating LLMs, VLMs particularly, on exams and stuff like that. And that was a lot of effort to collect the data. It was not a medical one, right? It was like
Yeah.
a more general specific. And the pipeline, when thinking about that, goes exactly as the pipeline when you think about doing like the research we do every day. You have to come up with the idea, why do you need this data set? How are you capturing? And then all the issues that emerge there. So yeah, I think developers who are interested in this aspect, they are more than welcome, I guess.
Okay, very good. So there was a question in the chat from Abdulhamid, is whether how were you able to combine datasets with different metadata?
Yeah, that was one thing. So what I mean, in this case, the metadata was pretty limited because we had only gender, we had age, and we had ethnicity mostly. And some of them had like different type of labels. So harmonizing that was not a big deal for us because it was okay. Some of them don't have ethnicity and they will not have. I mean, we didn't infer ethnicity to amplify the number of metadata available, right? So
Yeah.
in that sense, it was just like we created a table, we say these data have this,
Okay.
these data have that, and we like could do some subpopulation analysis depending on the available metadata we have.
Very good.
That was basically
Okay. Also on part one, before I move to the part on fairness, from Lydia. So Lydia is asking a question. I would like to develop it a little bit. So we work in a project, European project on breast cancer, and there we have breast mammography and breast MRI, and we have data from 6 hospitals: Italy, Sweden, Lithuania, Poland, and 2 in Catalonia, actually in Spain. And yeah, we had a situation where when one of the centers could not annotate their data sets, show us where the tumors are, perhaps segment the tumors. We were kind of stuck. We just didn't have the resources to pay a clinician to do it. So I don't know if you have any ideas how we could extend all the annotations we have in the other center to that last 6th center.
So that was segmentation. What kind of annotation was that, or was classification?
Yeah, it was Lydia, can you give more detail on the annotations you were looking for?
Yeah, sure. It was segmentation. Hello, Enzo. Nice to meet you. It was segmentation of tumors in mammography.
Okay. Yeah, I guess, I mean, depending on what's the quality of the other labels that you have, maybe you can think about training a model and providing some automatic segmentations for that, but really highlighting with a red label like this that these are automatic and these are like human generated, and even thinking about providing metrics that come together with the segmentations that you are generating. Now there you are talking about lesions, right?
Yeah,
Tumors.
my main issue here is that we have some clinical information for the lesions, and then we need to map the clinical information to the segmented lesion. So to do this automatically is quite challenging, of course, if there is no clinician verifying that this lesion it was the one that we got the biopsy from, etc. And we know the tumor subtype, for example.
So you have tumor subtype and all that, but you need a segmentation. Yeah, or I don't know, you can think about organizing a challenge around it and using that as the and so you get the power of the community to help you with that task as well. But yeah, I think it's pretty challenging. I mean, and the task we tackle here is a much simpler task than the one you are tackling, I think. I mean, we get pretty high dices here. That's why we had we say, okay, the model is working pretty well, let's go for a multicentric study like this, let's try to release all that. But in other cases where you get dices of 0.4, 0.5, 0.6, I wouldn't be happy to release that data set. So yeah, you have to see if the quality is up to standards, I guess.
Cool, thank you. I think Lydia will contact you offline, Enzo, perhaps. because we'd like to collaborate on this topic. I think it's very interesting. Okay, so let's talk about fairness a little bit. Let me see. Okay, let's talk about fairness. So first question I would have here is, yeah. So when we talk about fairness, in our field, we typically look at groups like gender or age. There was a question by JP in the chat. Is perhaps, what about looking at other type of unfairness, for example, to healthy versus unhealthy, or yeah, I could extend this question. What about a factor that nobody has thought of? No, that's actually very important, but we just either don't have the attribute itself in the data, or we just haven't thought about it.
Yeah, totally. Well, that happened to us with the AP versus PA I was mentioning here, right? So there was a subgroup that we didn't consider, and we found our model was much worse on that subgroup. But in this case, we had the metadata to perform the analysis, and we could do the subgroup population analysis. In case of healthy versus unhealthy, you could also have the target, and you can do that analysis and see, yeah, the model is performing much better on this group than other one. But to me, the most interesting question in that direction goes on what happens when we don't know exactly what's the reason why what's the reason that divides the people that are where the model is working well from not working well.
Yes.
And that's another type of unsupervised bias discovery problem.
Yes.
Like here I was talking about unsupervised bias discovery in the sense that I don't have labels, like target labels. But that's a type of unsupervised bias discovery where you don't have group labels, let's say.
Yes.
And yeah, we have been thinking about that. Actually, we have a paper we published last year at the ICCD that is called VIG bias, like visually grounded bias discovery, where you kind of use visual explanations to try to understand the failure modes, and then you use LLMs to describe these failure modes. So that's a way to go, like trying to use, let's say, okay, the model is failing in all these images. Why is it failing in all these images? What do these images have in common? And so you ask a model to describe them, and you get, all these people have glasses. I don't know. So that's why the facial recognition system is not working well, or something like that. This problem in the literature of computer vision is more known as slice discovery as well. We call slice, and it comes from the literature of spurious correlations, because sometimes the reason why the model is failing is because there is a certain spurious correlation as well. So this slice discovery thing, it's pretty, slices here are basically subgroups somehow. And you try to discover which is a slice in which the model is failing. So yeah, there is a whole bunch of work on that.
Sounds good. Last question, which is from Vin. She's wondering if the auto-labeling in CheXMask may have contributed to amplifying some unfairness or even creating some unfairness in downstream clinical tasks. What do you think?
Well, that's a very good point. I hope not. We did all our best to actually tell the world, okay, these are the subgroups in which the model is failing more and blah, blah. But yeah, it may happen. I don't know. I don't see a clear reason why that could happen in our case. But maybe. I mean, I'm not aware of all the uses that people are doing of that. That's why I always advise everyone working on these problems to try to do this kind of subpopulation analysis by themselves in the downstream tasks. That's the only way you can assure this is not happening.
Okay, thank you so much, Enzo. Thank you for everyone for joining this meeting. So again, the talk will be available soon on YouTube for everybody else. You can forward it to your colleagues. And yeah, thanks again, Enzo, and we look forward to seeing you in person somewhere this year, perhaps in Korea, no? Are you coming to Mikai?
I'm not going because my paper was rejected. Like our papers were rejected, so not going to Korea this year, I think.
Hope to see you another place. Yeah.
somewhere else.
All right. Cool. We'll be in touch with
Okay,
you, by
well,
the way, for
okay.
collaborations. And yeah, thanks again. Thank you, everyone.
Yeah, thank you for joining, everyone. I see many known faces, so thank you very much.
Goodbye.
Bye-bye.